Hello, I'm Teacher Ray, and today we will discuss genetic engineering. It is a process taking a gene from one cell, making some changes in the genes, and putting and altering genes back in the original cell. The organisms that have had genetic material removed or inserted in order to change a particular trait are called genetically modified organism or transgenic organism. Genetic engineering can be applied in many ways, and we will discuss its application in medicine. So we will talk about Humulin R. It is an artificial insulin. Insulin is a protein that controls the metabolism of sugar, and it is also one of the treatment of diabetes. Scientists were able to isolate the DNA sequence that regulates the production of insulin in the human body. After isolating, they were also able to develop a technique for splicing this segment into the DNA of the E. coli bacteria. The altered bacteria have now insulin manufacturing genes, and as they grow rapidly, they produce the protein of interest, which is insulin. It's a pretty cool process, and we will simplify the process in 6 steps. So the first step is to identify DNA from 2 sources. And in our example, the first DNA is from the human cell, which is the insulin producing gene. And then the second DNA is the plasmid from the bacteria. Now that we have our target, we are now ready to go to the step 2, which is how to get the DNA. The second step is the application of restriction enzymes to cut the desired DNA. The restriction enzymes are DNA cutting enzymes found in bacteria, and they are called the molecular scissors because they cut DNA at specific sequences. Once these restriction enzymes are added, restriction fragments separate with sticky ends at each edge. That is why we're able to get the human insulin producing gene and the plasmid DNA cut with the restriction enzyme. So we're ready to step 3, which is adding DNA ligase to bond 2 DNA to create a recombinant plasmid. The DNA ligase bonds the sticky ends produced by the restriction enzyme, and thus making it possible to bond 2 types of DNA to make a recombinant plasmid. Just a reminder on this process, you have to use one type of restriction enzymes, because if you use different types of restriction enzymes, they will not bond together because of the difference in the base pair sequence of the sticky ends. So now we're ready for the 4th step, which is inserting the recombinant plasmid to the organism. So once we're able to insert the recombinant plasmid, it is now called a transgenic bacteria. The next step is to allow the transgenic organism to grow in a culture, and as the bacteria grow rapidly, they also produce the protein of interest, which is insulin. And now we're on the last step, which is separating the insulin and the bacteria. And in this process, it is important to purify the insulin to make sure that the bacteria is killed and the insulin and the proteins are pure, so that it will not cause any allergy or reaction. It is now then packed at the laboratories and until it's ready to be used for human injections, since insulin is administered subcutaneously. Thank you so much for watching.